Good afternoon, everybody, and welcome back to the Why Now lecture series. Today's talk will focus on PET imaging. Here with us is David Izquierdo from the Integrated MR PET Imaging Lab. David is an expert in non-invasive molecular imaging applied to the study of brain and cardiovascular diseases. Thank you very much for coming, David.
Thank you very much, Valeria. It's a great pleasure to be here. Thank you very much for the kind invitation to join this Why and How. And I hope I can give you a nice intro to what PET imaging is. Just to be aware, if anybody is here, waiting to see some PET images from cats and dogs, this is not the right seminar for you. So this is going to be about positron emission tomography, and I hope you enjoy it. And obviously, my emails are here. If you have further questions, please don't hesitate and follow up. Throw me an email, and I will try to reply to your questions as soon as I can. Just to be aware, if you listen to some baby crying, I have a 4-week-old baby. She might have something to say during our seminar.
Thanks for letting us know. Okay. Whenever you're ready, thank you.
Okay, so welcome to this intro to PET imaging. A lot of my focus in my career has been on cardiovascular imaging. So some of the examples that I'm going to show you are going to be focused on the cardiovascular system. Although I've been working a lot in the neuroimaging arena, and I will try to show some of the neuroimaging images too, because I know most of the Martinos is mostly focused on neuroimaging, yes. But, just to be aware, a lot of my main focus right now is on the cardiovascular system. So I will try to make links to it too. And it will also be a good opportunity for some of you to see other areas that are beyond or below the brain in this case. So let's start. What is positron emission tomography? So positron emission tomography stands for, PET stands for positron emission tomography, which is basically a tomography, which is a 3D picture, 3D image of how the patient functions. So compared to MRI, which I'm sure most of you will know about by now working at the Martinos, MRI is an anatomical imaging system. In contrast, PET is a functional imaging technique. So we are not going to visualize tissues or organs or different type of tissue characteristics. What we are going to visualize is how your organs or tissues work. So how we're going to do that is by using a positron emitter particle. So radioactivity in this case. So the positron is the antimatter of electron. Yes, antimatter exists, and we use it in medical imaging, in this case for PET, to obtain PET images. So the antimatter, as you may know, is basically the positron is the antimatter of the electron, which means that they tend to join forces together. Yes, when there is a positron, it will try to interact with an electron, in this case within the body, because we have injected this positron emitter tracer inside the body of the patient, and it will interact with an electron within the body, and it will emit these 2 gamma rays. So the patient is the one that is emitting, unlike CT, which is there is an external source that emits radioactivity, X-rays in this case. In PET is the patient that is emitting. So a PET scanner is nothing else than a camera that is going to collect those emissions from the patient. So if everything goes well, we can end up with a very nice picture. In this case, this is a dopamine study. We are looking at the striatum in the brain of a patient. We can visualize, in this case, how this dopamine receptor system looks like and how is it working. So what kind of traces can we use for PET imaging? Well, there are many, many different options. This is a list of some of the radio traces that are currently available at the Martinos. Obviously, the most well-known is FDG. This is how PET positron emission tomography started many years ago. Around the 70s is when it actually hit into the clinical arena. So FDG is a fluorodeoxyglucose. Is basically a glucose analog. So what we are doing is using glucose, and we are changing it to use one of these positron emitter nucleus, which is in this case as an F18 radio tracer. So that glucose is going, for instance, to interact within any live cell in the body. The more energy a cell requires, the more it's going to use this sugar, this FDG, because this sugar is radioactive now. It will emit more from those cells that require more energy. Which cells require more energy, for instance, in the body? Well, cancer cells, for instance. Yes, they are incredibly active. They want to replicate themselves as much as they can. So they require a lot of energy. So they realize in the 70s, basically, that this glucose analog could be used very effectively to visualize cancer cells. And that's basically one of the main targets clinically of PET positron emission tomography. But in our center, we do more than that. Yeah, so we have a ton of other tracers to visualize many other things. A lot of them are actually for a neuroimaging. We can use tau tracers or amyloid tracers for a lot of these Alzheimer's and dementia diseases. We can see the neuroendocrine system. We can target the opioid receptors, the dopamine receptors. We can target the activated microglia for neuroinflammations, PBR28. We can target many different type of receptors within the brain, but also outside the brain. As I was saying before, I'm using cardiovascular applications as my main target for clinical applications. And for those, I will show you a couple of examples at the end of this talk on how we can use different traces that are not targeting any of the brain receptors, like most of us are doing at the Martinos right now. Also, this is a very good example. We can use even drugs, active drugs. In this case, is the temozolomide, which is a cancer drug that can be transformed into a radioactive tracer. At the same time that we are using the drug to treat the patient, we can also visualize by modifying the molecule slightly and adding this radionuclei with this positron emitter nuclei. We can use it to detect where the drug is going, where is it binding, where is it actually accumulating to treat, in this case, cancer cells. Yes. So how does it work? So there is basically 4 main components in positron emission tomography. So one, that is going to be a really strong magnet. And then, because the PET is about acquiring 3D images and acquiring actually a dynamic series of images, this is going to be very similar to what Cine is. And Cine is, the cinema is all about light, camera, and action. So those are actually the 4 main components: the magnet and light, camera, and the action. Let me show you how this works. So to start with, for PET, we really need to create these positron emitter particles, these radio tracers that emit positron. So in order to do that, because positron is a particle that is not very stable in nature, we need to create it. And in order to create it, we do that by using a huge magnetic field. In this case, what is called a cyclotron. We accelerate protons in the cyclotron, and we target on these cells here. Those are the target cells. We target nuclei of other compounds in order to remove neutrons from them and convert those old particles into new compounds, into new tracers that are going to be unstable and will be emitting eventually positron particles. So this is the way to create a radio tracer for PET emission tomography using a huge cyclotron. We have one on site next to the PET base in base 7 and base 6 at the center. And if you want to see one of the how is this working, you can send an email to Jacob Hooker and his lab basically, and I'm sure they will more than lovely actually welcome you to see how the cyclotron works, and I'll show you a little bit of what is happening behind those bars basically. Once we have the radiotracer, then we can inject it into the patient, and hopefully that tracer is going to be a biologically meaningful tracer. And by that, I mean that it's targeting some of the interest processes that we want to capture, yes? As I was saying before, we can capture some of the different receptors in the brain, dopamine, the opioid, the serotonin receptors, for instance, or we can make the tracers to bind into the tau tangles, so tau proteins, or to the amyloid proteins. So we can actually target however we want it. Yeah, we just need to create that tracer using the positron emitter nuclei that we just got from the cyclotron and combining using some radiochemistry in this case to create the product. But the important thing is once we have the radio tracer, it will be emitting, because it's unstable, it will emit a positron. And that positron will eventually travel a little distance. And there is a reason behind that, and I will explain you in a second. But once the positron has traveled a little distance, it will eventually meet with an electron within the body, in this case, because we have injected the tracer into the body of the patient. So that will annihilate both of them. Matter and antimatter love each other, and they annihilate. And they annihilate by actually producing two gamma rays, two photons, which is nothing else than light. It's light at a different frequency that we don't see, but it's light. So what the camera of a PET scanner is going to do is actually capture light in a very similar way of like our cameras on our phones or our photo cameras that we, the expensive ones that we have at home. They capture light that we see. A PET scanner will capture light that we don't see because it's a higher frequency, basically at the gamma ray frequency. So why this positron needs to travel is because most of the times the energy of the positron is higher than the energy at which it can meet and annihilate with an electron. Imagine that you are in a disco and you are dancing with a partner. You really need to have the same energy level to be dancing in synchrony, yes? So that's exactly what happens with the positron and electron. If they want to dance and annihilate each other in synchrony, they really need to have the same energy. And that's a very specific energy, which is 511 keV. If positrons have slightly higher energy, they will need to release that energy. And they release energy by moving in the same way as us. When we have a lot of energy, we go for a run or we do some sports and we release energy. Yeah. And that will have an effect, by the way, and I will talk about that. That's what is called, this distance is called the positron range. Okay, so we have the cyclotron producing radiotracer. We know that that positron from the radiotracer will combine with an electron and will emit these two gamma rays, these photons, this light. All we need is a camera. So this is an old scanner, a PET standalone scanner from 10, 15 years ago. Newer scanners are a little bit fancier and much more appealing visually and also professionally from the point of view of how they work. And in this case, it's not a PET standalone scanner, it's a PET CT scanner. And I will mention later on why we want to combine a PET with something, in this case with a CT, or in our cases, with MRs. So what is inside this PET scanner? Well, as I was saying before, it's just photo cameras. But those photo cameras are not like the ones in our phones, are special ones that capture that light that we don't see, but it's a high energy light, the gamma rays, yes. So at the end, what is inside this? Well, they are going to be these detectors, which are basically high efficient detectors to capture that light that is not visible and convert it into visible light. And those are called scintillator crystals. Okay, this one is what is called BGO. There are new ones based on newer components, but at the end they are basically they are scintillators. They convert light of high energy into visible light. And because that light that is converted is very, very tiny, very, very little tiny amounts, what we need to do is to multiply that. And the old days we used to do use the photomultiplier tubes. The problem with the photomultiplier tubes is that they are really efficient, they are amazing, really good, very little noise. But the problem is that they do not go well and their magnetic fields. And if we are going to be combining PET with MRs, which we love in our center, because that's one of the things that we are best of, then we cannot use these photomultiplier tubes. So there are new techniques right now that are using silicon, what is called silicon photomultipliers. This is the first generation, which is called an avalanche photodiodes. The new generations are called silicon photomultipliers, and they are much better. They operate really fast. They have what is called this time of flight acquisitions, and they have a much better gain, and they produce more light. So they are much more efficient than the old generation of APDs. So we have the tracer. We know that they emit gamma rays. We have the camera. All we need is to collect the action on the movie. So we are going to place a patient in the scanner. In this case, this is a brain. We inject it with the tracer that we want, targeting the right receptor or molecule or organ or tissue, whatever you fancy. You are the ones deciding what you want to target and what you are going to be injecting to the patient. All we need to do is to capture those gamma rays. So at some point, one of those positrons will be emitted in one part of the brain, in this case, and will emit these two gamma rays. Those two gamma rays will need to arrive almost in coincidence, basically at the same time. What is the reason behind that? Well, these gamma rays travel at the speed of light. So the distance traveled by both of them is going to be, traveled by both of them is going to be very similar, no matter what distance are they apart from the detectors. So because they are traveling incredibly fast, yeah. So it's only a few picoseconds difference for a 60 to 70 centimeters scanner, wide scanner, basically. So there is some fast electronics behind those detectors, those camera detectors, that are telling us if one detector is hit at the same time than another one. And then we will know that there is what is called a coincident event. We know that a positron was emitted, a positron was annihilating with an electron, and these two gamma rays happen almost simultaneously. And this is called a coincident event. Yeah? So this is only from one positron emission. This will happen millions and millions of times, and we will collect all these events in coincidence. And that's what is called raw data on the scanner. It's collecting all these coincident events on time, so taking the time as a stamp and saying which detector was paired to which detector at which time. And we can acquire this for minutes or even hours and have not only a single shoot of what is happening at the patient at one single time point, and we can do it at several time points. In general, at a large interval, up to 2 hours or even more sometimes. So that's basically the action of the movie. So now that we have all this raw data with all these coincident events, how can we produce an image? Yeah? And this is part of how we can reconstruct an image. This is the image reconstruction problem. So in this case, I'm putting here an example, obviously a simulation of a letter I. And the letter I is full of positron emitting matter. So basically at this radio tracer, the rest is basically empty. There is no radio tracer, so nothing is going to be emitted from there. So only positrons are going to be emitted from inside the letter I, and it will emit these two gamma rays by annihilation within electrons within that letter I. So how am I going to reconstruct it? Well, basically, I'm just going to use those lines, color those lines, and I will show you in a second how, but I can basically by now just color those lines, joining those two PEG detectors that were hit, and I start accumulating millions of that, like this. Okay, so a lot of millions of these positrons are annihilating with electrons within that letter I, the patient in this case, it could be the brain. And by accumulating those lines of response, by putting some gray level to every one of those lines of responses, and I can accumulate millions of those responses, and at the end, I will start obtaining my letter I. This is what is called a back projection. Okay, But because this image is really horrible, you know, nobody works with this type of images, we do more things behind that. And part of it is, first of all, I'm not going to be assigning to every voxel, which is basically any one of these squares. A voxel is a 3D version of a pixel. You know, the pixels are the camera elements on your phones. A voxel is the 3D extension of that 2D camera. So instead of assigning exactly the same value of every voxel that crosses that line of response that is hitting from this positron annihilation with electron, I'm going to be assigning the value relative to how much of that voxel area the line is crossing. So for instance, this voxel here is crossing only through one of the corners, so it's a very little area that is crossing. I will color it with little gray. The ones that cross a lot, like this one, cross from one side to the other, it's almost basically the whole voxel, it will be colored darker, and so on. So I will have a gray of colors, basically, through that line of response, yes? And again, I am going to do that millions of stuff with every one of those lines of responses. And at the end, that is going to be giving me a slightly better version of the image, but still not perfect. So let's suppose that instead of my letter I have more of a brain type of image. It's not, obviously it's not a brain, but you know, a similar type of shape with a couple of structures that could be, for instance, brain tumors. Yes, this is what is inside the scanner. If I start adding those back projections with those you know, different gray areas on each part of the voxels that are crossed by the line of response, I will end up with this version of the image. It's already very similar to the original image that is inside the scanner, but it's not too good. And that's because I haven't filtered it yet. So I can add to the back projection image, which is this one, I can add a filter, in this case, a high resolution filter, basically, that is going to be, you know, erasing all those low frequencies and actually make my image much better. And why am I erasing the low frequencies? Because we know that, you know, from all those lines of responses, those areas that are very uniform in intensity are very likely not going to be contributing to the image. It's only areas where it's, you know, there are changes in the image, high frequency changes will be contributing. And that's basically what is creating this very similar image to what in reality is inside the scanner. And this is called the filter back projection. This is the gold standard, by the way, of the image reconstruction in positron emission tomography. But this is the old system. This is the old way to do it, even though it's the gold standard, and it's the only version that is still unbiased in terms of PET quantification. Nowadays, we are doing iterative methods, which are much faster and less noisier. And the bias is nowadays very little compared to the FPP. So we are accepting these iterative methods, which give us better solutions to the image, and you know, in a better way, basically. So we can obtain images that are qualitatively and quantitatively pretty good. So we said that, you know, these two gamma rays will need to arrive in coincidence. So as we can call it a coincident event. And that's basically what we are going to store as the raw data. But there are many different things that can go wrong while collecting this data, yes. And in fact, you know, there are, you know, many different things that can go wrong. It is called randoms, scatter, attenuation, background noise, normalization, because the different lines of responses, different crystals, the detectors have different efficiency. There is the death time. Whenever there is a lot of those gamma rays hitting the detectors, the electronics might not be too fast to acquire all the data. So there might be some pile-up effects. So I'm not acquiring all of them. Sensitivity effects, for instance, in this case, every plane of the image might not have exactly the same counts, and then we need to reassign how much count are equivalent to a certain dose, and that's what sensitivity is, or decay. Let me show you some of those examples and what they do and how we can correct for that. So we said that, eventually we wanted to have the events in coincidence, but sometimes one of the gamma rays might not be hitting the detector, either because it's going outside of the camera or because it's actually crossing the detector without being detected. That can happen. So if at the same time there is another event like this one, where one of the gamma rays hit one detector but the other one is lost, those two, if they happen at the same time, again in coincidence, they can be considered as a really coincident event, and we will assign a wrong line of response. This is what is called a random coincidence. Okay, only when these two events are coming from two different positron emitter sources, but are hitting the detectors exactly at the same time in coincidence, and that's what is called a random coincidence. If they are not hitting the detectors at the same time, they will be called single events, and we will record the single events, but we will discard those events because we don't want to process them. Okay, so that's the random events. We can correct from those, by the way, by using a delay coincident window, because these random events will be similar across time. If I acquire a very delayed window, I can know how many of those coincident random events will happen, and eventually I can assign the same value across my window of interest, which is the coincident window, which is only a few picoseconds in these days. So scatter, what is scatter? Well, scatter is, when one of the gamma rays that is going to hit one of the detectors, all of a sudden deviates because of its interaction with matter. Again, those are gamma rays, these are photons, and they interact with matter. In particular, they interact through Compton scatter based on the electron density of the tissues. So for instance, bone has a higher density than water-based tissues. Bone has a higher chance to scatter and to absorb the events, which is coming later, which is called attenuation, by the way. So when this happens, we will have still in coincidence those two detectors, but they are not the right ones assigned. And instead, we will incorrectly assign this line of response. Okay. So how can we correct for that? Well, we can use, in this case, in these days, we can use Monte Carlo simulations, knowing what is the electron density of the tissues. In this case, we will need to know what is the pseudo-CT, in this case, the pseudo-CT image of the patient, yes? And I'm coming to this in a second, because this is related to the second part, which is the attenuation. So Compton scatter is when the matter deviates one of the gamma rays out of the scanner, but actually both of them can, or one of them, sorry, can be lost. And then we only have one single event that we said we are going to discard anyway. So at the end, we end up with no line of response. And that's what is called attenuation. And attenuation can happen again because of Compton scatter, or because the gamma ray is completely absorbed within the matter, so within the tissue of the body. Okay. So how important are these corrections? Well, attenuation is the largest correction for PET imaging, and it can be really huge. And this is an example in the whole body on how much it affects. So you will realize that before attenuation correction, a lot of the tissues that are very low attenuating, like lungs, for instance, have much higher intensity than other tissues that are more dense, like muscle, for instance. And then you will realize that inside, deeper inside the body, we have less activity than it really it should be, because more inside the body there is more dense matter around to get those gamma rays out of the body. So the more the chances to get either absorbed or scattered. So to correct, again, we were saying that we need electron-based density. CT X-rays are electron-based density images. So we can use a CT image to correct for attenuation correction and then for scatter, because we can use that information to simulate what the scatter was. So this is one of the reasons why PET and CT can pair together. And there's another reason that I will tell you in a second. So what is the impact of attenuation in the brain? This is how big it could be. So if you don't do attenuation correction properly in the brain, you can end up with these really biased images. So quantification here will be really tricky, and you will be losing a lot of information basically. So correcting for attenuation and doing it properly is incredibly important. Another big important correction is decay. So what is decay? Well, decay is basically how much that radioisotope basically is decaying, is disintegrating with time. So in terms of the initial activity, if we have 100% activity at time 0, the decay of the radionuclide is going to be going down following an exponential with what is called a half-life, which is the time by which the tracer, the initial activity has gone to half. That's called the half-life of the radiotracer. So this follows an exponential with this half-life as part of the exponential terms, okay? And that's always the case for every positron emitter radiotracer. So what is changing from an F18 radiotracer to a carbon-11 radiotracer to an oxygen-15 radiotracer is that this half-life is going to be very different. So F18 has a half-life of 2 hours. So basically, this the activity of the tracer will be half by 2 hours. A carbon-11 will be 20 minutes, which is basically this curve in here. An oxygen-15, the half-life is 2 minutes. But copper-64, for instance, it can be up to 12 hours. And there are other tracers with even larger half-lives. So obviously, the shorter the half-life, the less radiation a patient will have, but the more tricky the acquisition will be, because we will have less and less time to acquire all the images that are necessary to understand what is happening behind the brain or behind any tissue or organ of interest. So how good is PET imaging to detect molecular processes? Well, PET is actually the most sensitive molecular imaging technique that exists right now. And here are some examples, and courtesy from Simon Cherry from UC Davis, of how little radioactivity needs to happen within the body to be able to detect. And this is important because you will probably have noticed, and I should have said before, PET images are blurry, and that's because the resolution of the PET scanners is poorer, much poorer than the MRI or the CTs. Yeah, an MRI we can go even now below 1 millimeter voxel resolution. For PET images, the resolution is around 3-4 millimeters, 5 sometimes. When you're very good dedicated PET scanners, you can a little bit lower than 3, but it's still not as good as an MRI image. But the important is that it's so sensitive that we can even detect objects that are smaller than the resolution of the scanner. And this is highly important. It's not all about the size of the object that we can detect. It's about how, what is the sensitivity of the scanner to be able to detect it. So in this case, for instance, if there is a point source type of lesion, for instance, inside the body, like a cancer lesion that is really, really concentrated, like a point source, is only the detection limit is around 9 becquerels, which is basically 9 disintegrations per second, which is incredibly low. That translates into around 250 cells concentrated in the body that need to be, for instance, if we are talking about cancer, that need to be cancerous to be able to detect them. And that is incredibly small. That amount of cancerous cells cannot be anatomically visualized with MRI or with CT. And that's the importance of why PET is so critical for the use of in oncology patients, because it's able to detect incredibly early when there is only a few, dozens of cancer cells already in the body. We can be able to detect it. So, what are all the factors that affect, in this case, spatial resolution and PET positron emission tomography quality? But there is many. Is the positron rate, yes, how much that positron is traveling because it's high energy before it annihilates with an electron. Well, that will have an impact, yes, and because we are assigning into a slightly different source that line of response what is happening from the origin of it, yes. I don't want to enter here, but that will increase a little bit how much we are able to detect smaller objects. Non-collinearity. So we said that those gamma rays need to are happening in opposite directions, exactly in opposite directions, but sometimes it might not happen exactly in opposite directions. Yes? There might be a small, very tiny variation of the angle. The larger the scanner field of view, the much larger the deviation, that tiny angle will hit the detectors. And that non-collinearity will increase again will actually decrease the resolution, will make the object blurry. The detector side also impact, the higher the detector size, the blurrier the images. The depth of interaction. So basically it is important within a camera detector, whether it's impacting at the on the very beginning or at the very end of the detector, because that will increase a little bit the distance from the light on response. Detector scatter can also happen inside the detector, and it can actually deviate out, and that's also increasing. Sampling, so how much are we going to sample the image, and what is the voxel size that we are going to use, or even reconstruction parameters, yes? So all those are affected. But if all go well, eventually we should have a nice PET image. And this is basically my take-home message for today, is how PET works, all the components that need to happen together to obtain a good image. Is just we need to have a huge magnet, the cyclotron, that produces these radio tracers. Those positron emitter nuclei will emit a positron that annihilates with an electron and emits these two gamma rays in opposite directions, hopefully in coincidence, and that is basically what it makes positron emission tomography. We need to detect that light, those gamma rays, photodetectors, which are these scintillator crystals with these either photomultiplier tubes or silicon photomultipliers in these days. And then we need to process those lines of response, those detectors being hit together, constructing these lines of response, and accumulate them into what is called an image reconstruction. Yes. So whenever everything goes pretty well, we have amazing quality images, PET images. This is a dopamine receptor study for raclopride. And look at this image. This is fantastic. Yes, we can actually look at two different regions. And in this case, I'm looking at this is part of the striatum, and these are the radii here. And in this case, if you look at one single static image, like a camera shot, what can happen, and I don't know if it happens to you, but it happens to me all the time, and also other people, is that sometimes the picture that you take, you don't have your best face, yes. And it happens a lot of times. Sometimes you think, the face is weird because you were, having a bad day, or it catch you in the wrong moment. And sometimes it might be the reality, yes. So in order to distinguish that, instead of taking just one single picture, we can take a lot of them, and we can take a video out of it. And that's exactly what PET is very good at. We can take pictures, videos of, in this case, the dopamine system, but of any tracer that you want, and start looking at what is happening. If we only observe, in this case, the static images that we were looking at those areas, we could have thought that they were having very similar uptake. But the reality is that those two regions behave very differently. And we can construct these time activity curves, this variation of the uptake in a certain region, and follow it in time. And that will give me the dynamic nature of the tracer within that region. Yeah? So how the radio tracer is behaving with time. And those are called time activity curves. And we can use those time activity curves to perform what is called pharmacokinetic analysis. Yes, and there is a lot of mathematics behind it. I'm not going to go through this. That's another why and how, by the way. You can ask. There's great people behind: Julie Price, Kristin Sander, Hannah Hansen. You can ask them to give you. I'm sure they will give you an amazing why and how on pharmacokinetic analysis. There's a whole theory behind this. But all I can say is, there are mathematical models that model the biology behind that. Because the tracer, once it gets into the body, it needs to, it's on the arterial compartment, it's first on the blood pool, and need to cross, for instance, in the brain, the blood-brain barrier. For instance, that will be this compartment here. And it can also cross back into the blood pool, and you can add more and more compartment of these compartments, modeling, for instance, the specific receptors versus the non-specific receptors, or it's called the free or unbound tracer. You can have even more if there is a non-specific versus a free that is different. So you can increase the complexity. Obviously, the more complex, hopefully the more accurate it is, but the more challenging it is to fit all these parameters. Yes, those are all differential equations. They are difficult to fit, and the more you are adding, probably the more noise you will have, and the more challenging it will be to have a nice fit. So you need to compromise how much of of that information you want to acquire, how little number of parameters you want to fit, because that will help you to get better statistics versus how complex in reality your biology is. So how different those pharmacokinetic maps look versus what is the raw data? Well, it depends, it varies on tracer to tracer, and it varies on the disease, not only on the tracer, but also the disease model, but it can be very different. So this is the raw data for a MISO rat model, stroke model. And this is what the raw data looks like, and these are what these K1, K2, K3, these pharmacokinetic parameters look like. Very different, yes, and that's because these K values, these K maps, they are answering specific biological questions. Yes, K1 is basically the extraction rate from the blood pool, from the arterial blood pool into the tissue. So in this case, the non-specific tissue, the free compound, and then K2 and K3 are, one is the return, and the other one is the one that goes into the specific compartment, etc. But what I mean is that pharmacokinetic analysis is very powerful. You use the whole dynamic, the whole video picture of your PET emission tomography data, and you avoid those funny pictures that I was showing you before of having one single shot. And that's a beautiful thing that we can do with PET imaging. It's complicated, but it's doable. So, as we were saying, PET scanners are now combined with CTs most of the time, but in our center we don't do the CT, and I will explain you why. Why is that? Well, one reason is because PET imaging is a lower resolution, and we want to have a matching high resolution imaging technique. CT is very good on that, and it's also providing the anatomical images behind the functional from the PET. Yes, so CT is an anatomical imaging technique like MRI. Instead that it involves more radioactivity, because it's X-rays basically. You are throwing X-rays to the patient, and in a tomographic version. So why do we want to combine those two? It's not only to just fuse those anatomical images. It's because sometimes a single point of view can be very deceiving. And let me show you a very clear example about that. This is a picture of Prince William taken on one of his venues in London. He was coming out of the car, and one of the photographers took this picture. And everybody was thinking, oh my God, how rude he is. And again, taking one single picture from one single angle can be very deceiving. The reality is that there were other photographers around it, and they took the same picture at the same time, in this case, from a different angle. And then they were, we were saying that in reality, he was not doing anything rude. He was just explaining that there were three things that he was talking about. Yes. So why combining two scanners? Is because then we can get not only one single point of view from the PET or the CT or the MRI, but from both combined together, and better visualize, better capture what is happening behind the disease that we are investigating. So though the biological process behind that, we can capture from different angles. And that, in many cases, helps understand what is happening. So in our case, I'm saying that, instead of a PET CT, we are using a PET MR. Well, there's a good reason behind that, is that MRI is much more versatile than CT. We have many different types of controls that we can use in MRI. And MRI, on top of it, it can provide also functional imaging techniques using fMRI, which, our center is pioneer, actually, 25 years ago. So we can not only combine the MR and the PET and fuse them together to specifically colocalize the signal from the PET on the MR and know exactly in which anatomical part the signal is concentrated. But in our lab, we are actually using the MRI, the synergy with the MRI and the PET, to improve PET quantification. And we are doing it in different ways, and I'm very quickly going through some of these slides. For instance, using for attenuation correction. We were saying that before, to use attenuation correction for PET, we can use the CT image. So instead, because on a PET MR we don't have a CT anymore, we need to create that pseudo CT. So this is an MR image of a patient, and this is a method that we came up with some years ago that is currently the one that is being used in many different parts in the world, by the way, and is the current standard and in both base 6 and base 7. It's called the SPM8 pseudo-CT method. And from this MR image, we can produce this pseudo-CT automatically, and it's almost identical to the real CT of the patient. So we can basically simulate that attenuation CT-like image and use it for correcting for attenuation correction for the PET images. And we can do that very successfully. But not only that, we can also recreate artifacts from the patient due to implants. In this case, it's a hip implant on a patient. This method was developed by Nicolau, one of the postdocs that was here in the past with us. And he created a method that using some MR techniques that are slightly less affected by the artifacts of the implants to recreate what's the actual attenuation correction of the implant, which is huge, by the way, because they are normally titanium, which are very electron dense, basically. So that means that they attenuate a lot. So it's even higher attenuation than more, much higher. So they will induce a lot of bias in PET quantification if we don't correct properly for this. And that's important in a lot of the pelvic imaging studies, prostate or ovarian cancer, or GI cancers, a lot of areas that cover, around the pelvis with these huge pelvic implants, this will have a huge effect. So another area is motion, and this is very important in many cases. Every single patient that you put on a PET scanner will move. No matter how good the patient is, no matter how much you ask them not to move, they will all move. Some will move more, others will move less, but they all move. The effect of motion is basically blurring your image. It will not only blur your image, but it will also reduce the quantification because of the blurring effect. And that has a huge impact if you are trying to obtain accurate quantification. So that also happens in the MR images, yes. But we can correct for that in MRI by using, for instance, EPI volumes, very fast volumes that can be acquired every second or every 2 seconds, and we can register those volumes together and correct for that. So the new MP ranges, for instance, which are volumetric images that are acquired in 5 minutes, use these EPI navigators to reposition the image and acquire a noise-free image. So we can use the same information from these EPIs in order to correct the PET image. So this is a PET image from a patient, from a volunteer with motion. If we correct using those EPI navigators, we can actually visualize much better the cortex of the patient. But motion is not only tricky because it also blurs the image, it's also because then there is a mismatch between the image, the emission image, and the attenuation. And this is something that can be really tricky and can actually mimic some of the diseases like temporal dementia. So in this case, because of the emission and attenuation mismatch, the patient moves to one side, in this case to the right of our image, the left side of the image becomes less intense. And that is very similar to what happens in some of the dementia, of the lateral dementias. And this is basically a phantom mimicking exactly the same effect. If we move the phantom to one side because of motion, simulating motion, we have exactly the same effect. So this is something that can mimic a clinical outcome. And if we don't realize and we don't correct for that, it can be really impacting the patient. There is a PhD in our lab is actually using motion, but not from the brain, but from the heart, because I was telling you that I'm using a lot of cardiovascular applications, and in this case using the MR motion from the CNA images to derive cardiac function, and using that now to get into the PET images and correct PET images from cardiac motion. This is what his next step is going through right. And finally, the last slide on corrections basically is in partial volume affairs. That's Kevin Chen, who was another former PhD student in the lab. He was working on reconstructing the PET images with less blurring effect, yes? And that blurring effect is especially variant. If this is the center of the scanner, there is a circle, and I'm dividing that circle into quadrants. This is the upper right quadrant of the scanner. If we move off the center toward one side, you will notice that this blurring moves towards that side. If you move up, you will notice that that moves also the blurring towards the upper side. If you move diagonally, the blurring will also move diagonally. So there is a spatially dependent blurring effect. We can use MR information to deconvolve that blurring, which is called this partial volume effect correction. And that was done by Spencer Bowen too. And Kevin came up with, in a collaboration with the UC Davis people, came up with this MR prior reconstruction. This is still not fully implemented. I know some of you will come and say, hey, I want that image on the right. Yes, this one here, which is nicely reconstructed. But, there is still some pros and cons on doing this. Yeah, some of them is like, well, we might be introducing some signal that might not be really present. This is one of the things that we're actively looking at right now. So the good news is, there's a lot of reconstructions and things to process with talking about positron emission tomography, but luckily we have a lot of tools already available that make all of this automatic or semi-automatic just on the push of the button. Brain PET, in basics, we have this mass immune tool. On the MMR, we have this MMR tool with our triplet reconstructions behind that too. And that will actually facilitate a lot of these attenuation problems. Scatter and motion now these days are now fully automatic inside these reconstructors. So that's the good news. So very quickly, a couple of examples of non-brain oriented PET positron emission tomography using non-conventional tracers. And this is a tracer developed by Peter Caravan. It is a fibrin binding tracer that binds to fibrin, and fibrin happens to be the main component of blood clots. So what you are seeing here is a blood clot on the lung. And it's so tiny, we can see it very clearly on the PET image, but it's so tiny that it doesn't not even appear on the MR image. And no matter how good your high resolution MR image is going to be, you will never detect this because this is really incredibly tiny. It's subclinical, there is no clinical symptoms. This patient was absolutely perfect, yet the patient still had a small a small blood clot. And this is the beauty about having these 2 point of views. We have the MR in this case and the PET. On the MR, in this case, they happen not to be visualized. On the PET, yes, but combining them together, we get the best of both worlds. We have the high resolution image to know exactly where that clot is located within the lung, and we could intervene. In this case, there was no need because it was, nothing was happening to the patient. But if it needs to be, it will. So when is a problem is when the live clots can be life-threatening, and it can be producing heart attack and strokes. In this case, this is a blood clot. Let me have it here. This is a blood clot inside the heart in the left atrial appendage, which is one of those cavities like an appendage, like in our GI system has an appendage, the colon basically has an appendage. The heart has also a small appendage, which is, part of our evolution. And that tends to accumulate clots within certain populations, like atrial fibrillation patients, where the heart is very asynchronously and fast pumping. It can detach those clots and become life-threatening. If they fly out into the brain or into the heart, again, into the coronaries. So again, joining MR and PET together, not only to provide nice anatomical confirmation of the position of the blood clot, but also in this case, using functional techniques from MRI, in this case a T1 map, we can actually confirm to that there is a blood clot because it has more methemoglobin in this case. And the last example is using MSTAT, which is a HDAC receptor radio tracer, which is the Martinos stat developed by Jacob's group. We can eventually visualize the effects of epigenetic factors affecting the metabolism in the heart. And this is something we've been looking at in collaboration with David Sosnovic and Jacob, basically looking at how the heart is changing, likely due to those epigenetic factors that affect HDAC, HDAC expression, and that downstream has an influence into protein formation and gene expression, basically. And we are still working on this, and the images are absolutely stunning from the point of view of using nicely 4D reconstructed images, basically cardiac-gated images. If you want to know more, this is probably the Bible for PET imaging tomography. It's a book from Michael Phelps, which is not the swimmer, by the way. It's another Michael Phelps, which was the inventor of PET positional emission tomography. And if you want, just Google this book, PET molecular imaging and its biological applications. It's fascinating. It's really easy to read, and it's fantastic. A lot of acknowledgements, obviously, from my lab, from the integrated PET lab, from all the group in here, but also a lot of the collaborators: Peter, Jacob, David, Ignor, Julie, David's lab, Bruce, obviously, Christine, Hannah, Xiaojing, Christina, for all these amazing discussions, every week for the big PETMAR group. A lot of acknowledgements, obviously, to them and to the NIH for funding, obviously. So if you have questions, I'm all ears. You can also drop me an email if you need to